the exam, I would at least read over that to know the numbers. You have chromosome 8, chromosome 14. So basically, this example, Burkitt's lymphoma, is going to be involved with two genes, MYC and IgH. MYC is an oncoprotein. So you make a bunch of MYC, that's going to mean what for the cell? I have a bunch of MYC. Cell is going to divide, which is cancer. Hey, look at that, right? This is Burkitt's lymphoma. So, normally, should MYC be highly expressed or low, like now, in us right now? Should we have high levels of MYC expressed or very low levels? Very low. Moderate to low. What about IgH? IgH is one of those immunoglobulins, right? So IgH, is that normally going to be active or not active? And once again, it's a lymphoma, so it has to do with T cells. This is actually, it's a T cell. It's going to be proliferation, I believe, of the T. It's one of the blood cells. I think it's the T cells. IgH normally is pretty active. Why? Because immunoglobulins, right? We need to be making those constantly to be active because you're constantly making new immunoglobulins to fight possible cancer or possible, not cancers, antigens, right? Is that what it's called? Bunch of antigens going around. So normally, IgH is relative, has a, maybe not high, but there's a moderate level of IgH expression because these cells are constantly going to be making different forms of immunoglobulins. These are B cells, right? Not T cells, sorry. These are the B cells. So they're constantly making IgH, IgA, all the different IgGs, and they're going to be slightly different to try to bind to any possible antigen. Normally, though, that's okay. IgH should be moderately high. MYC should be pretty low, if non-existent, because if I have a lot of MYC, I'm going to be constantly dividing. What happens in Burkitt's lymphoma is I get a translocation where that MYC gets put pretty close to so here's what we have. We have this chromosome. This is my IgH. And this is basically going to be, we'll leave that there for now. I'll explain it in a sec. And then we have this. This is my MYC. What ends up happening is I get this translocation where I have this. Now, these two genes aren't snacked together. This is not a fusion protein. What happens is, now when I go along and I transcribe this, I'm going to first make IgH the protein, and then I make MYC the protein. And the reason I have a relatively high level of IgH, you have this upstream part of this DNA is going to be very open. It's going to have response elements that T transactors are going to bind to. So this is always open, always going to be moderately open. Whereas here, this is all very, very close. Little to no MYC. So because of this translocation, IgH is already made at a moderately high level. What's going to determine whether this is at a higher level? What comes upstream of it? Therefore, now it's in the same area. I'm making a lot of IgH. I already was. This is no different. But now I'm making a lot of MYC. And what does MYC do in the end? Increased cell proliferation. Now I get this cancer type. So that's what's going on. IgH is highly active. That basically what that little ball represents is a highly active area of transcription going on. MYC normally is all closed off on its chromosome. Because of this translocation, now it's in an area that's highly active for transcription. Therefore, I make a lot of MYC. And that's what happens with Burkitt's lymphoma. That's that right there. All right, now we're doing fusion genes. So instead of this example, think of like this one gene getting smacked into this gene. So now when I'm transcribing it, I'm making this like Frankenstein monster protein because it's going to have two different genes put together. So for example is the NPM ALK fusion. Basically what's happening here is that normally I think the ALK has to be phosphorylated for it to be active. And ALK is going to be responsible, I believe, for it's going to be for pro-proliferation. So it's going to cause cell division. ALK needs to be phosphorylated. ALK is part of a signal pathway, don't worry about it, but when there's a high phosphates on it, it's going to be active and it's going to lead to cell cycle. Because when this thing gets fused to this protein, this gene in particular, I make a protein where NPM basically has phosphates on it, or phosphate-like things on it, like what? Glutamate and aspartate. And it makes the ALK thinks it's active. So the ALK Frankenstein is active because there are phosphates on it. So you make NPM ALK one long monstrous protein. The NPM part of it has glutamates or aspartates on it, and the ALK thinks that the ALK part thinks it's active. So therefore, it then shoots down that entire cycle, and I then get cell cycle progression. Other example is BCR-ABL. This is for chronic myeloid leukemia. So I would know the disease that's going to be due to this fusion. And in particular, Gleevec is a drug that treats chronic myeloid leukemia. So Gleevec is a drug that treats this BCR-ABL fusion. And once again, this drug in particular, it's only targeting BCR-ABL. Do my normal cells have this fusion going on? No, my normal cells are totally fine. So this is another good drug with very few side effects because it's only targeting this Frankenstein. It's not targeting the normal cells. I have no idea what the slide he's trying to say. I think it's looking at different ways you can have fusions. But if you have a question about it, ask him tomorrow because I think he's gone after that, right?
Yeah.
So, up to this point, we talked about how mutations are going to lead to these different cancer phenotypes. Either we can get an actual amino acid substitution, we can get a translocation, fusion example of that. We can get amplification, right, where I just increase the number of times that gene is in the DNA. Or really, it could even be changing the transcription levels of just the one gene. We could have RAS in terms of it being, RAS can be due either due to having way more of this, getting amplified of the gene, or mutating this gene, this RAS by itself, so making it more active on its own. So it can be, it's not always one or the other. So, gene amplification. Now we're going to talk about ways and technologies, Blank's favorite thing that we can look at it. You've got HSRs, homogenously stained regions. Think of it kind of like, you know when they did the staining of the chromosome for your ideogram? It's kind of like that. Not really, but what they can do is they can take a cell and they can go through this technology and have parts of the DNA stained. Every part of the DNA that's the exact same and repeated will be stained one certain color. It's like a fluorescence. And it'll be like that way all the way through. So we can do that and look at if there is an area that's stained longer than it should be, we can assume there's an amplification there. If normally I just have like my little pinky stained, but on another cell type, on the other one it's actually a lot longer like that, that whole area is repeated, I have an amplification. It works the opposite way too. Where if I have a deletion, I'd have a pinky normally, and I have like nothing over here. So it can be both for amplification or for deletion. Double minutes, these are small portions of DNA that they're containing amplified amounts of the same gene. However, these are actually like, I don't understand how it really works, but they're not actually in the DNA. They're like a little outspurt, like they're not in the whole chromosome. They're like little offshoots of it. So in the picture, you can see here, the really, really, really small things, they're going to be those double minutes, whereas these big, sort of like these blobs right here, those are the homogeneous, the HSRs. So those are the ones where it's that repeated region of that amplification. The double minutes are much smaller little pieces, much smaller little things. Comparative genomic hybridization, definitely know this. Definitely. It's nothing that new. Basically, we can only use this if I want to compare levels of a gene, or not levels, the amount of the gene I have. So whether a gene is amplified in a cancer cell type or it is deleted in a cancer cell type. It does not look at mutations. It doesn't, well, this is type mutation. It does not look at amino acid changes. This does not look at base pair mutations. This does not look at different transcription levels. We're looking just at the DNA itself, whether the gene is amplified in a row a bunch of times. So if you have a disease that's due to increased expression of a single gene, would this show anything different? No. because it's still one gene in the cancer cell, one gene in the normal cell. What's different is these other transactivators and shit up here that's allowing this to get transcribed more. And we can only use this if there are different numbers of genes in the original, in the DNA itself. So what we do is we have to pick colors. We're going to pick colors, and we're going to basically then look at this cell type versus the other cell types and see if there's a color difference between them. So what's going to happen is you've got your normal so you have your patient. Let's say they have breast cancer. It has not metastasized. The breast cancer is only in the breast. That means, are my other cell types mutated? Do they have the mutated BRCA2 or any other mutation? No. Well, hopefully not. So you've got your BRCA2 mutated in the breast cancer cells, other cells of the body that don't have the BRCA2. This doesn't work because it's a mutation. So pretend it's a random disease where it's due to increased amount of the protein. So let's just say it's gene A. So here's what happens. Your bad cells, they have 5 of gene A. This gene A is going to be an oncoprotein. So I have way more gene A, that means I get way more cell division. Your happy cells, they only have 1 copy of gene A. So what we do is, I need to take a cancer cell from you and a normal cell from you. And what I'm going to do then is, I'm going to break up all the DNA into, I basically know what gene I want to do, but you can do it off in the entire genome. We know exactly what to add to break it up in the correct way, where now I'm going to have my blown up DNA, both the healthy DNA and the bad DNA. I then add fluorescent dyes to each of them. So I'm going to add fluorescent dyes to this one, all this DNA, the good DNA, and dyes to the bad DNA. All the bad DNA gets 1 color dye, all the good DNA gets another color dye. I think, I want to make sure I did the same thing. Okay, cool, because then it'll make it make a little more sense. So what is it? Red and green. So normal is red. So all of these, the little subunits that we broke up, they get red. Everything from this cell gets green. Then what we do is we throw them together and we see what color we get out of it. So obviously red and green together, I think it gives you yellow. I don't remember my primary colors, but I think that gives you yellow, right? No one knows? Okay. So you've got the normal DNA, the cancer DNA, from the 2 cells from the same patient. Okay? And what you did is you blew up both the DNAs, all the normal DNA, all of it, you gave them the red dye. All the good DNA, or the bad DNA from the cancer, you gave them the green fluorescent dye. You do this for all of the, basically all the DNA, all the genes, not just for this area, because we don't actually know what area it is, but we do it for all of it, and we put that together, and then we hybridize them. We take these we blow it up and make them go from double to single strands. I then hybridize them. So I'm taking DNA from 2 different sources, one source being the normal DNA cells, one source being the mutated DNA, cancer cells, and I put them together. And when you put them together, these 2 individual sections then, since these are complementary base pairs, they're going to match up. And that's why I get this color. So if I did red and green, and I bring them together, you end up with yellow. What that would tell me is, this gene in particular is not amplified or reduced. It could be mutated. It could be due to changes in transcription level, but the actual physical number of the gene, how many times it's there, is the same. It was 2 in the healthy cell, and it's 2 in the cancer cell, because the color tells me that based on the fluorescent dyes. If we have normal and cancer, and we have a lot more of the cancer cells, we end up getting a lot more, when these come together, a lot more of these green dyes. I do not know how this works. I've tried looking at it, because if you think about it, there's only one of these strands. How do all 3 bind to this one? I have no idea. It's probably in an article somewhere. But just note that even if they're unequal amounts, that's still good for us. Because in this case, I have way more of the green than the red. What I may suspect is that these really don't bind to each other. They're binding to something that we made that then collects all of the same ones together. And that'll tell you if it's red or green, but Blank doesn't care if you know that. So basically here, I only have 1 from the original, but I have way more green, so what color am I going to see at the end result of this? at this location? I'm going to see green, not yellow, not red. So if I see a lot of green, that tells me there is an amplification of this gene in this cancer cell line. If I see a bunch of red, what does that tell me when they're combined? That means a deletion has been occurred, where I have less of that gene in the cancer cell type than I did in the normal cell type. Once again, this only gives us info on the physical number of the gene, of that same gene in the DNA. That's the overall color scheme we talked about. Microarray, you can also do this with CGH, where basically you do the normal and the cancer, denature it, give them the fluorescent dyes, and throw them on the same chip. And then, we're not sequencing shit, we don't care what the sequence is right now, all we care about is the number. So if I throw them on the chip, and I get more green or more red, that tells me, oh, that means there's way more of this green in the cancer cell type versus the red in the normal cell type. Same general idea, except we're doing it on a chip, not doing it with whatever that other sequence is going to be. Once again, colon cancer. Definitely know this from the previous again. HNPCC, the hereditary non-polyposis. So there's no polyps, but it's colon cancer. What's the first thing that occurs? We lose MMR. In particular, one of these parts of MMR, MLH1 or MSH2. These are lost first. These play into MMR. MMR, all the repair mechanisms, it's not one protein, right? It's a host of proteins that all together work. If I screw up one of them, overall the entire repair pathway may be screwed up. However, just because I have this mutated, this doesn't mean that I'm going to have unrestricted cell growth. This just means I don't have this repair mechanism. In the case of HNPCC, we lose MMR, either the MILH1 or the MSH2. Where's the next one? Oh, okay, we'll get there in a second. Hold that thought. But yeah, this is just looking at percentages. No, don't worry about it. I mean, Blank could throw that on there, but he has other more important things to test you on. So I'd say this is very, very low priority. But just be aware of that this is a hereditary disease. So there are higher percentages in families versus a normal population. And how could we have initially figured this out? Maybe doing those genome-wide association studies, right? And looking at percentages between the different population sizes. So yeah, once again, MMR mismatch repair. Keep in mind, MMR uses an exonuclease, whereas NER and BER use an endonuclease. Exo means what? It's got to come from the end, like a Pac-Man. Go back. Endo, it can pop in wherever. An endonuclease is different than an exonuclease. So HNPCC, we've defective with that sort of mismatch repair. In the case of HNPCC in particular, we first lose MMR. After we lose MMR, the gene that is very often mutated is TGF-beta receptor. It's the tumor growth factor beta receptor 2. We lose that receptor after we lost the MMR. MMR gets screwed up, and then when TGF-beta R2 gets mutated, I can't fix the mutation. And this, for some reason, this gene in colon cells is much more susceptible to mutation than other cells in the body, and we're not entirely sure why. So we lose MMR. That's bad if we lose it anywhere, but in the case of in the colon, when we lose MMR, this is a very high chance of getting mutated, very susceptible to it. And the end result of this is that this receptor is one of the receptors that actually leads to the RB pathway. So by having this screwed up, the RB pathway is not going to be correctly functional, not going to operate correctly. End result is going to be going off from that down there. So the net result of this is what happens when you normally have it. It binds to this and you get decrease in the proliferation. However, now we don't have the receptor binding, so even though the signal is here, it can't bind. So our receptor is screwed up. So therefore, we get to this increased transcription of proliferation, and we get the increase in the cell cycle progression. We're talking about this exo versus endo. Breast cancer, BRCA1, BRCA2, or BRCA1, BRCA2. BRCA1, we haven't really talked about it much. It's important for cell growth. It's going to be dealing with things like the kinetochore and other things, some parts of DNA replication and repair. But the one we talked about in this class extensively is BRCA2. That's for recombination repair. It's that one plus RAD51. Important for remember, both those help stabilize that strand invasion. Remember we get like that weird triple helix at one point where you are using the other sister chromatid set of the homolog for that. That's all that's talking about right there.